Hello everyone, welcome to the last part of this series, how to perform the molecular docking using the using the Autodock Vina. In in the previous video, we have seen how to prepare the protein. After that, how we have prepared the ligand using the different tool. After that, in the previous one video, we have actually performed the auto molecular docking using the Autodock Vina. And now in this video we will going to see how we are going to be analyze those results which is generated by the Autodoc Vina. So if you see over here we have the different ligand poses has been generated, that out of the 9 exhaust units that we have been selected while performing the molecular docking. So, in order to analyze those results, I'm going to use the Discovery Visual Studio, which is a better option that I feel to analyze those results which is generated by the uh, Autodoc Vina. So, to perform to analyze those results, first thing that you will have to do, you will have to open the ligand. So over here you can either upload the ligand that you have been selected into the PDB-QT. So if you see, this is the protein that we have selected in order to perform the molecular docking. So in the hierarchy, if you see, we have only chain A and the protein group that we have. Okay. So now I will have to add those all the poses of the ligand which is in the protein that we have been selected. Okay, so the first thing that if you see the ligand one, the out ligand one will be the highest scoring. If you see the -7.1 in the log file, if you see, so I'm not going to be add uh, all the ligand poses. So I will just add the first one, which is the high, which is showing the highest score over here. So that is the -7.1, which shows the highest scoring over here. That is the out ligand one file will be there. So in order to add that particular file, first thing you will have to do, go to the file, then um, click on open, then to go to the location. So my location will be the C docking, and after that you will have to select as all files, yeah, at the bottom. So if you see, all the files will be reflected. So I will just going to add the out ligand one will be there. So click on that and click on open. So once you click on open, so if you see the ligand, this is the ligand uh, that actually dock with the protein of choice that we have been selected. So what you will have to do, so in the hierarchy, click, uh, right click, then click on the copy. Okay, so once you copied the ligand, then go to the protein hierarchy, then again right click and paste it over here. So if you see in the same hierarchy, we have added our uh, ligand. So this is the ligand that we have added over here. So in order to to see the docking that has been generated and the docking interaction between this particular ligand of choice and the protein, so select the out ligand, then go to the view interaction. Under the view interaction, you will have to define the ligand that you have been selected. So click on the out ligand, then click on the define ligand. So once you define your ligand, then go to the ligand interaction. So if you see, you are able to see the interaction between this. So now we have the certain option over here to display the aromatic environment, the hydrogen bonding, then the charge, then the hydrophobicity, then the ionizability. So these are the few options that we have. You can select this particular option. You can select, you can also add the receptor over here. If you see, the receptor has been added over here, and you can see the different interaction over here. If you added the aromatic and the hydrogen bond environment, you can expand it, you can the contract it. So these are the few options that we that will be available over here. So if you see, we have expanded it to, it will automatically get expanded. And the whichever option that you wanted to be select, and you can the those options that you can select over here. So in order to generate the 3D diagram of this particular interaction. So you will have to get it into the publication quality. Okay. So to get this particular diagram into the publication quality, so I'm just deselect as hydrogen. So if you see, you can clearly see the interaction. So I will just, uh, get the position in such a way that in the similarly we can take the snapshot of that. Okay. To save this particular 3D interaction diagram, the first thing that you will do, you will go to the script, under the script, go to the visualization and select the publication quality. So this is the publication quality that we have been selected over here. And now, in order to save this particular file or this particular position or this particular snapshot in 3D interaction, so go to the file, then click on save as. Under the save as, you can select as a image file. You can select as a image file, and 3D interaction diagram you can give it over here and save. So ultimately, so if you see, the 3D image has been saved over here. If you see in the similarly the position that we have been shown over here. So this is the uh, 3D interaction diagram that we have selected over here. Now the second, in order to view the 2D diagram of interaction, click on the uh, 2D diagram. So if you see, you can see all the interaction of that particular ligand of choice. The compound which is get interacted with the different amino acid, along with the interaction bond which is presented into the different color code over here. And those color code, this 2D diagram you can like use to uh, to use, or you can copy this particular 2D diagram into the research work that you wanted to be copied it into. So in order to save this particular 2D file diagram into the image file, the same option that you will have to be go to the file, then click on the view, then the automatically it reflected as a image file, then select as a 2D, and then you can select the file. So if you then click on the OK, the pixel that actually on the file format. And after that, if you see, the 2D diagram has been saved over here. So this is the 2D interaction diagram that has been reflected over here. So this is for how to save 2D and 3D interaction of those compound uh, or the 2D and 3D interaction of protein and ligand complex. After that, the next thing that you will have to analyze the result, the name of those amino acids, then the category and the those you will have to analyze along with the bond length has been formed by that specific amino acid with our ligand of choice. So there was, if you go to the there was a at the bottom there on another one hierarchy will be there. So if you click on that up arrow, so if you see, that will ultimately get expanded, and after that go to the non-bond. After that, in the non-bond, you can see those interaction over here, the name of amino acids, and the distance, that is nothing but the length of those bond which is formed with the ligand of choice. The category of bond has been reflected as the four hydrogen bond and one hydrophobic bond has been formed by the ligand with the selected protein. And if you see, the type of bond has been formed, whether it is the hydrogen bond, whether it is a pi alkyl bond. So these are the different category and types which is formed along with the length and the name of those amino acid which is get interacted with the our protein of choice, sorry, ligand of choice in the uh, docking that we are performing within the Autodoc Vena. So this is all about to analyze those results which is generated by the Autodoc Vena, and these are the several steps which is involved from the start to the end to perform the uh, molecular docking. So so this is how you are going to be perform the molecular talking. If you have any questions, you can reach out to in the description. I have provided the email ID of our services, and that you can reach out to us anytime whenever you needed to perform the molecular docking. So thank you for joining in, and uh, don't forget to like, share, and subscribe our YouTube channel. Thank you, everyone.